Hi. In class, we're doing a pond water ecology lab. And in this lab, there's a couple things we're trying to do. But before that, as far as safety goes, the main thing is with slides and cover slips. They're glass, and we just want to make sure that we try not to drop them. And if we do, we don't cut ourselves. So try to hold your slide two hands if you're carrying it around, or hold it over your lab tray, because if it falls on that, it probably won't break. Second thing is with your location, to make sure you're not accidentally bumping into someone. Stay at your lab station. If you need to go somewhere, just ask. That pause causes you to take a look around and be aware of your surroundings, less likely to bump into someone. And then with the microscope, today we're just using scanning and low power, so you shouldn't have any problems of the microscope hitting the cover slip or the slide. What we're doing here is we're taking a look at a couple pond water samples. Here's an example. This is from a backyard pond. You can see there's a whole bunch of stuff floating in it, and you need to make a couple initial observations. You want to look at it, see what it's like. If there's anything moving around, great. See if it has an odor, and record that in the qualitative data section in your science notebook. Then we start identifying organisms. And with that, we're going to put it under the microscope, go to scanning power, get it in focus with the coarse adjustment, move it around, and find anything we can, and use one of several field guides, maybe this one here or this one here, to try and identify based on the size and shape of the organisms, and then get a count. And what that looks like in our notebook is this. In our notebooks, we're working on page 20 and 21. The question we're trying to answer with this lab is which populations of organisms live in local pond water. The procedure we will write out, and it's really just as the last slide. We're making some initial observations. We're then using scanning and low power to find any organisms that we can identify, and then we're getting a count. And then down here in the observation section, we are going to create a data table, and you need at least 2 samples. There's several up here, and you could bring in your own. You want to write down the location where it was from, your initial observations, and then down here is really the organisms present. In your online notebook, you could click here to get a copy of the guide or one of the ID guides, and we have those in class. And then eventually we're going to make some sketches up here, and those sketches are of the things you see. So try and sketch them. If there is some color, add it. And then eventually we will write our conclusion down here about this ecosystem. Hope that answers your questions and outlines what we're doing for this lab. If you do have any further questions or need any extra help, please request to come in during intervention block or block 3, and I'd be happy to help.